This conference will now be recorded.
First, I will explain you the GoToMeetings app. So on the GoToMeetings app or web page, you can find 4 icons at the bottom of your center of the screen. That is mic, camera, screen, and leave. Mic, click on that to mute or unmute yourself. I request you all to stay muted while our speakers are presenting their research work so that it doesn't cause any disturbance to them. Camera, you can use this button to share or unshare the webcam. If you have the option to share the webcam, you can do it. Otherwise, it's completely your choice. Screen, you can use this button to share your screen. This is the main part during your presentation. After I call your name from my side, I will be making you the presenter. Only I give the permission, you will be able to share your screen. So once I make you the presenter, you can click on the screen button and you will find a pop-up window to share your screen. And the users using the MacBook, you need to give you need to give extra permissions in your system preferences. And the fourth icon is leave, where you can use this to leave the session. On the top center, you can use the options to change your display settings. At the top right corner, you will have a 3 icons, people, chat, and settings. In the chat option, you can chat with me or other participants. For the other settings, for the settings you can use this button to check for the settings of your speaker and microphone. So as mentioned, kindly remain muted during the presentation so that our presenters don't get disturbed. And for any questions, you can ask the questions or you can send your questions in the message box too. Well, let's not take much of your time. Finally, I would like to request you to give a brief introduction about yourself before starting with the session so that we can know about you virtually. So as for today's webinar, the first oral presentation was by Dr. Sunita Singh, but as we can see that she has not yet attended the webinar. So we'll go to the other talk from Dr. Gispi Gullo from University of Palermo, Italy. And the title of the talk is Fertility Preservation. Hello, Dr. Gispi. Are you ready with your presentation?
Yes, I'm ready for the presentation.
Yes. So you are the presenter now. Please click on the screen icon and start presenting.
Yes, but I'm trying to charge the presentation. So the template.
Yes, we can able to see your screen.
Yeah. Just a second. Mhm. Sorry, I have to remove the chat and the screen because I didn't. Just a second. This one. So I'm just opening the PowerPoint. Can you
Yes,
see my
so
slide?
yes, you can make it a full screen. Yeah, perfect. Please start presenting.
Yeah, thank you very much to invite you to this really interesting and eminent conference, the International Webinar on Clinical Research and Trial STEM. What we are going to speak is about the fertility preservation. So I understand the burden. So the next slide, please. So I try to, okay. So first of all, I want to clear that I don't have any potential conflict of interest in this presentation, and everybody know about fertility, especially women, because I'm an IVF consultant. During the time when the age growing up, the fertility is going down in an inverse relationship between the two principal hormones, FSH and LH. The principal hormone is the follicular stimulant hormone that is going up, and the AMH, the anti-Mullerian hormone, is going down. So what we suggest usually in our discussion with the couple, that these patients and this couple, we suggest to plan a family when they are early, younger than no more than 30, 32, because we know that the most cause of abortion is the chromosomal errors that can happen during the mitosis and meiosis differences. So I want to show you this slide, how in Europe, especially in my country, in Italy, the couple that start to think to a family and the baby born is dramatically going down. So as you can see in the next slide, from the unification of Italy and today, the reverse pyramid, you can see how how the percent of baby for each family is about 1.2, no more. So when we speak about fertility preservation, we start to speak about cancer, because we usually suggest to preserve the fertility when there is, unfortunately, diagnosis about cancer. And in this case, we can see the percent of the most cancer in women, breast 31, you can see 17%, and decrease to melanoma, thyroid, as you can see. So what are the factors that influence the fertility? Congenital and hormonal disorders, lifestyle, environment, and sexual transmitted infection. And when we speak about female fertility, the age, of course, is the most factor. The lifestyle, the smoking are other factors that have a big influence, and the exposure to pollution. But we don't have to forget the sexual transmitted infection, especially HPV, chlamydia, trachomatis, and that's all. So we usually refer to our guidelines about the two principal international society, the European, the European Society for Reproductive Medicine and the American Society for Reproductive Medicine. I want to remember you how genetic factor, infection, pelvic surgery can dramatically affect the ovarian reserve. So the time is really important. When a couple comes to us, it happened 1 hour before to me, and we discuss about unfortunately diagnosis of cancer. Earlier is a better chance. The time is really important. What I mean, the couple come to our visit, we need to start an ovarian stimulation in one week, 10 days. So what we do, we give gonadotropins, hormone, and we have to apply a random start protocol. So we have to start in every moment of the cycle, because these patients, you can understand, that have to go under chemotherapy, radiotherapy, that they are gonadotoxic, so they need to stimulate the ovary. We have to pick up this retrieval all the oocyte we can and to freeze all. What we do usually is important a correct counseling, because these are patients that need also psychological support. And as we can see in the other slide, the fertility preservation in patients undergoing gonadotoxic therapy is important to freeze their gametes, especially the technique that we usually use is the vitrification system. Vitrification system that is more fast than the previously used technique that was slow freezing. What I mean that oocyte cryopreservation is a consolidated technique for the ovarian stimulation that can be performed in patients with hormone-sensitive cancer. And we are referred to, for example, the breast cancer. The breast cancer, where we use the letrozolo. Letrozolo is a molecular that is an aromatase of third generation, as you can see in this slide, and high selective, and have the possibility to give during the administration of gonadotropin, FSH recombinant, or for alpha follicular recombinant, have the possibility to decrease about 90% the percent of estrogen that can be bad for the breast. This is a publication that I did the last year on Fertility Street. It is a really good journal where we compare, and I want to show you the vitrification system. We did two groups. The first was to compare the embryo quality, pregnancy, and outcome in between the two vitrification systems. The closed system that is more safe for us, what we demonstrated in an aseptical way, and to the open vitrification that is the most used. And the other group, we compare the outcome embryo quality to between vitrification and fresh oocyte. So we found the same result. Chemotherapy, what can induce? Ovarian reserve depletion, menopause, ovarian damage. So as reproductive medicine, in my opinion, and in the last 5 years, all the reproductive medicine, vitro fertilization, reproductive endocrinology at all, is going to to study other factors of embryonic metabolism. And we start to speak in the last year, as you know, maybe about transcriptomics, genomics, proteomics. So what I think, what I want to share with you, my thing is that all the reproductive medicine is going versus a personalized medicine. What it means? That the actual, present, and future of the reproductive medicine is going versus a tailored medicine, a specific therapy for each. You don't have the same therapy for all the women, for all the men that need to preserve, of course, of the men that they have to freeze their semen. But there is the therapy that is better for you, that is different for another person. And just to show you this other publication, the gender gap, where you can see how the diagnostic therapeutic process of infertility campus helps to eliminate the gender bias, indicating how to design equal access to infertility diagnosis and treatment. So what I want to suggest to you, to suggest your work and take on this message that to improve, to suggest people to do also the social freezing, because not only for cancer, patients can freeze their gametes when they postpone to plan a family, and they can freeze their gametes at 25, 23, 28, when the quality of the eggs and the semen is good. So they will use, when, for example, they will be 40 their gametes to have a baby. And I suggest to freeze their oocyte, not embryo from the couple, because you know sometimes not all the couples stay all together for the rest of their life. So it's better to prefer the oocyte than embryo. Study the biochemical markers, FSH, LH, anti-Mullerian hormone. It's important to study also the thyroid. And today, to conclude, I want to tell you how the indication to cryopreservation is increased. It is increased because the demand of social freezing is also going up, because it's growing the egg bank, and this represents an example how today, for example, the pollution, also chemical radiotherapy, are really problem to reduce the ovarian reserve. Thank you very much, everybody. Thank you.
Thank you, Dr. Gisby, for your presentation. So any questions from anyone? So no questions. Thank you once again for your presence and presentation.
Thank you, you too.
Yes. So as we can see, Dr. Sunita has arrived to the webinar. So we will go with her presentation. So our next presenter is Dr. Sunita Singh from Navyog Kanya Mahavidyalaya, Lucknow, India. And the title of the talk is Exploring Ayurvedic Medicinal Herbs for Their Antiviral Potentials. Hello, Dr. Sunita. I'm going to make you the presenter.
Yeah.
Yes. So please click on the screen icon.
Okay. Yeah.
Yes, we can able to see your screen. You can make it a full screen.
Okay. Okay. Yeah.
Yes.
Is it visible?
Yeah, perfect. Please continue with your presentation.
Okay. Thank you. First of all, I would like to thank the organizing committee of International Webinar on Pharmacology and Toxicology for giving me this opportunity. The topic which I'm going to discuss is exploring Ayurvedic medicinal herbs for their antiviral potentials. As we all know that for last 2 years, we are combating with a viral infection, that is COVID-19, caused by a virus that is SARS-CoV-2, which shows that the viral infections are not very easy to handle and shows the complexity of this virus. And we have seen that there is a debate around the vaccination drive, also the vaccines which has been developed in a very short time, which forces the scientific community to go beyond the available options like antiviral drugs and vaccines, And in this case, medicinal herbs are found to be very potential candidate for developing drugs related to the antiviral potentials. In the 21st century, pollution, unhealthy lifestyle, environmental toxins increases the risk of diseases. The side effects, overuse of allopathic drugs are also a major concern. And we have seen a trend of a green consumerism where people are now going shifted towards the products of natural origin as well as towards the traditional medicine system, especially during the COVID-19 infection. We have seen people are using different formulations derived from Ayurveda, especially in India, for treating various infections, especially in case of milder, we can say, symptoms. There is a lot of literature where the traditional medicine system has shown their potentials in treating different types of infections. Although there has been also drawbacks like proper rigorous clinical trials are needed. I'm going to discuss what are the problems which are facing these traditional medicine system. In 2013, WHO developed and launched WHO Traditional Medicine Strategy for the year 2014 to 2023 and emphasized to integrate traditional and complementary medicine system with the conventional. As we all know that the still having so much advancement in the scientific tools, still 80% of the population are using traditional medicine system for primary health care, which is a very large ratio and which shows the importance of traditional medicine system in which herbal plants and their bioactive components played a very important role. This is a figure of a circle of life for achieving universal and quality life. Especially in case of traditional medicine system, we can see the major proportion is given to the preventive measure as well as the healthy lifestyle, which shows that the traditional medicine system works on the root cause of that disease and avoid the different factors or the cause which can responsible for developing that disease. Whereas in case of allopathic system, we have a special treatment technique, we have a portion of medicine, surgery, diagnostic techniques. So these are the some we can say functions that allopathic system use. Traditional medicine system, either it is in Ayurveda or it is Chinese medicinal system or Campo, they all work on a basic principle that to avoid that factors which are causing that disease and works on that root cause and to give a holistic approach to the immune system of that person, and which strengthens the immune system and provides the strength to fight against different types of infections, not only viral, other infections also, like we have bacterial, fungal, etc. We have hello, I'm audible? Hello?
No, that's okay. You can continue. Sorry.
Okay, sorry. It is estimated that 25% of the medicines on the global market are synthesized from natural products. And in anecdotal evidences, we have many evidences regarding medicinal claims of different plants. Of the estimated 3 lakh plant species that exist worldwide, only around 15% have been evaluated for their pharmacological activity, which is a very less in number, and more than 2/3 of the world plant species have medicinal values. So it shows that the plants are the potential candidate for this drug development processes. During the year 1981 to 2019, Food Drug Administration has approved more than 1,500 drugs, either made from unaltered natural products, botanical drug mixtures, derivatives of natural products, synthetic drugs with pharmacophores from natural products. These are some of the numbers approved by FDA, which shows that natural products, either in the direct form or in the mixture, like we have a synthetic drugs with natural pharmacophores. Why there is a need of alternatives? As we have seen, actually this generation has seen something like COVID-19. We have seen mass casualties, unpredictability of availability of vaccine, time lag between vaccine development, emergence of mutant drug resistance, prohibitive cost of available drugs. These all drawbacks, we can say, or the challenges has forces the scientific community to go beyond these options. That's why we are now, or the scientific community are questioning the herbal plants. If we talk about the viral pathogenesis, simple steps are involved. Everyone knows. I'm not going into deep. Like entry into the host, local or primary replication, viral spread, effect of the virus on the host cell, host immune response, fate of the virus after clinical recovery, and virus shedding into the environment. So these are the basic steps of viral pathogenesis, how this virus infests into the host cell. When we talk about how these antiviral drugs work, it basically inhibits the viral entry. It helps in viral uncoating, interferes with various synthesis process like transcription, protein expression, inhibits the viral genome replication. So these are the mechanistic pathway used by synthetic drugs. When we came into the antiviral herbs, how they works, they basically target the enzymes that are involved in replication and integration of virus into host cell. In case of DNA viruses, restricted entry of viral particles into host cell, it inhibits the viral replication into the host cell, destruction of viral envelope, and reverse transcriptase, which plays an important role in replication of RNA viruses. So they restrict the activity. So these are the target sites where these antiviral herbs and their bioactive components work. This is one of the figures which represent the various medicinal plants and their possible specific inhibitory sites to act against SARS-CoV-2. We have heard about these terms, especially for last 2 years, like ACE2 receptor, that is angiotensin converting enzyme receptor. We have transmembrane protein serine factor. So these are some of the active sites where if we block or restrict the activity, we can indirectly stops or inhibits the progression of that viral infection. So while choosing the plant, the main purpose is to target these sites. Like in other slide, I'm going to discuss that in case of ashwagandha, they work on specific sites, and molecular docking approach has found that different plants and their bioactive components work on these targeted sites and helps in the we can say helps in the restricting the progression of viral infection. So I'm going to discuss about 3 plants, that is moringa, ashwagandha, and licorice. Moringa, its botanical name is Moringa oleifera, family is Moringaceae. Each part is beneficial for the medicinal purposes like leaves, flowers, pod husks, stems, seeds. It is basically commonly called as a miracle tree because its all parts has medicinal values. It is commonly called as drumstick tree, horseradish tree, shobhanjana in Sanskrit. It is native to sub-Himalayan tracts of India, Pakistan, Bangladesh, and Afghanistan. Various parts of this plant, such as leaves, seed, bark, fruit, and flowers, has been advocated for nutritional, medicinal, and industrial values. According to Ayurveda, the leaves of the moringa tree prevents 300 diseases and combat malnutrition, especially among infants and nursing mothers. In Siddha medicine, seeds of moringa are used to treat different dysfunction-related issues. And in African traditional medicine, the plant is popularly used for influenza viruses and different type of viral infections. The general health benefits of moringa involves it reduces blood sugar, lowers cholesterol, reduces inflammation, fights free radical, it treats depression, anxiety, and stress. It has antibacterial and antifungal properties also. The antiviral properties of moringa. This table shows that against different types of viruses like HIV, that is human immunodeficiency virus, herpes simplex virus, different extracts of this moringa shows very good activity in inhibiting the viral properties, like and the mode of actions involves inhibited it, like in case of HIV, water and methanolic extract inhibited the viral replication. In case of hepatitis B virus, the extract using water and hydroalcoholic solvent, it decreases various factors which are related to inflammation. In case of Newcastle virus, methanolic extract used as a immunobooster. In case of foot-mouth disease virus, the leaves of moringa is found to target the protein enzymes which are responsible for viral application. So These are some of the mechanistic pathway how these moringa leaves and their extracts has been used for their antiviral properties. If you talk the chemistry inside moringa, the different parts contain bioactive compounds and nutrients such as phenolic compounds, fatty acids, carbohydrates, fiber, minerals, vitamins, and functional peptides with a wide potential to be used in food. This is the structure of some components present in moringa, that is ellagic acid, beta-amyrin, epigenin, benzyl isothiocyanate, myrcetin, quercetin. So these are some of the bioactive components present in moringa. And if we talk the futuristic aspect of this moringa oleifera fruits and different parts like phytoconstituents, which I have described. So future drugs can be derived using these phytoconstituents, and the molecular docking has also supported the fact that they has a targeted action against in case of SARS-CoV-2 virus. So molecular docking has supported the antiviral properties of moringa. The next plant is ashwagandha. Botanical name is Withania somnifera, family is Solanaceae. It is commonly known as Indian ginseng, comparable to Chinese ginseng, winter cherry, ajagandha. It grows prolifically in dry regions of South Asia, Central Asia, and Africa. In Ayurvedic system, it is the most used herb for the brain tonics or rejuvenators. The roots of the plants are categorized as rasayanas, which are reputed to promote health and longevity by augmenting defense against diseases. The general health benefits of ashwagandha involves it helps in reducing blood sugar levels, it improves bone health, it helps to fight cancer, improve thyroid functions. The study shows that the components, the chemical components like withanolides, the major constituent of ashwagandha, is responsible for its antimicrobial, anti-tumor, and immunomodulating properties. Ashwagandha increases the production of nitric oxide, which is responsible for activating the macrophagal actions of the immune system and improves the ability to ingest invader foreign cells. So indirectly, it strengthens the immune system to fight against different types of infections. It possesses the power of combating disease like herpes simplex virus, which has been used by African tribes. It also helps in lowering the inflammation by reducing the number of C-reactive protein in the body, which lowers the chronic inflammation and helps the immune system to perform effectively or efficiently. It plays a vital role in host rejection of both tumors and viral infected cells. This is one of the collaborative study done by one of the Institute of India and Japan, where they have used the two components of ashwagandha and propolis, that is withanol and caffeine acid, caffeic acid phenylethyl ester, and they found that in case of SARS-CoV-2, they blocks the active sites which are responsible for the progression of viral infection, like withanol and CAPE blocks the M protease enzyme that is responsible for viral application in cells. So they blocks that the activity of M protease, and also withanol itself restrict or inhibits the property of transmembrane protein serine factor, which is responsible for the viral entry into the host. So it shows a positive side of these herbal plants, which are supported by docking study also. The cellular effects of withanolides, the constituent of Withania somnifera, involves antioxidative, anti-inflammatory, apoptotic, anti-proliferative. These are some of the chemical compounds: withanolide T, withaferin, and withanol found in ashwagandha. The probable role of Withania somnifera at various stages of COVID-19. Why I am citing COVID-19? Because it is a very current stage and we are still dealing with this COVID-19. That prevention of viral entry and replication, stabilization of immune homeostasis, elevation of hyperinflammation, antipyretic effect, organ protection against inflammation. So these are the probable role how these Withania somnifera can work if it goes from this herbal plant stage to the drug development stage. The next plant is licorice. We have heard about licorice candies also. The botanical name is Glycyrrhiza glabra and the family is Fabaceae. the root part is very beneficial for the medicinal purposes. And recently I have read an article about that they are using for, they are also evaluating its anti-cancerous properties also. So which is a very good sign beside antiviral and other infections. Now anti-cancerous potentials of licorice is also evaluated. Commonly, it is known as Mulathi sweetwood, and in Sanskrit, it was called Yashti Madhu. More than 1250 preparations are derived from this licorice and described in Ayurvedic system. The plant is commonly used for treating common cold, sore throat, vomiting, acidity, gout, weakness, joint pain. And during this we can say during this COVID-19 infection, people has frequently used this giloy, ashwagandha, licorice for different throat-related infections. The general benefit involves combats digestive ailments, fight respiratory infection, protects your liver, aids reduction, eases menopausal symptoms, fights arthritis symptoms. But beside all these advantages which I have cited or given in different literature, but still we are in very initial stage, and many research and clinical trials and safety assessment is needed, because the notion which is are common in the people mind or consumer mind, that the thing which is natural has no side effects, but it is not true. Like in this slide also, avoid taking large amounts of licorice for longer than 2 weeks can be harmful. So we have also reported different types of side effects of these natural plants. Like in giloy, we have seen that they have a bad effect on kidney if it is used in overdose. The antiviral properties of licorice involves, we have two major components, glyretinic acid and glycyrrhizic acid. Glyretinic acid, that is GL, against viruses like hepatitis C virus, herpes simplex virus, influenza virus, hepatitis, duck related virus. It affects released, it is effective against these viruses, and the mechanistic pathway involves it affect release step while infectious hepatitis C virus particles are infected cell, establish a resistance state. It reduces the factors which are responsible for inflammation in the body. It inhibits viral particles. It activates lymphocyte proliferation. Whereas glycyrrhizic acid, in case of rotavirus and human respiratory syncytial virus, it prevents viral attachment, internalization, and stimulates interferon secretion. So these are some of the basics mechanistic pathways how these antiviral properties of licorice has been established. Ellagic acid, beta amyrin, Chrysine, myrcetin are some of the major component. I think this slide has been come. Present and future prospects of this field, as I have already told, it is in a very initial stage. It is a very good approach. We can say that WHO establishes the Global Center for Traditional Medicine System in India recently yesterday or one day after yesterday, that WHO director has inaugurated the center in Jamnagar, Gujarat, India. Which shows that they are also recognizing the potential of traditional medicine system, and it is a high time to integrate this medicinal system, traditional medicine system, with the conventional medicine system or the modern science and technology. In India, ganjuveer and gingiveer is some of the drugs that has been developed from the plants. Ganjuveer is derived from Desmodium gangeticum plant, and it is found to be the first and only patented Ayurvedic antiviral medicine, which can treat common cold to COVID-19 like infections. And gingiveer is a herbal mineral tablets having major constituent of ginger and clove. The problems which are need to overcome before going to the drug development stage, like quality control issues, like starting from identification of a plant, which involves proper knowledge regarding the plant is needed, processing issues, harvesting time, post-harvesting storage. Biopiracy, lack of documentation is one of the issues that has to be addressed. Clinical trials, rigorous clinical trials are needed to assure the safety, we can say, parameters. Irrational use, as I have already also told about that, the notion about natural things is risk-free, which force or which can easily allows the person to use in a very irrational way is also one of the problem. Research and development related issues, as we need a proper infrastructure, especially to dedicate for research and development. Other issues involves, like we have pharmacovigilance issues, like safety and toxicological issues, and how they are going to be when we applied in vitro and in vivo studies are needed. Infrastructure is needed, which is a part of research and development. And administration, there is no regulatory authority, although especially in India, a department is now dedicated to the this traditional medicine system, that is Ayush Ministry, which is involved in promoting the different aspects of traditional medicine system of we have Ayurveda, Siddha, Unani, homeopathy, naturopathy. So now, especially in India, government has recognized the potentials and doing work in this field, and to go from this general health benefits to a very specific, or we can say in the drug development stage. These are some of the references. So I would like to end my presentation with a quote, that is, Mother Earth's medicine chest is full of healing herbs of incomparable worth. And now it is a very high time to recognize this worth without exploiting this this chest of medicine chest that has been given by our Mother Earth. And I would also like to wish a happy Earth Day to all of you. And thank you very much for the patient hearing. Thank you. Thanks a lot.
Thank you, Dr. Sunita, for your presentation. I'm
Thank
sorry
you.
for the delay. Yes, so anyone have any questions?
If anyone has any query, they can mail me also. So
yes, so if you have any questions also, you can mail us. We will forward it to Dr. Sunita. So thank you once again for your presence and presentation.
Yeah. And I would like to also thank you also for the cooperation that your team has given me.
Thank you very much. So as our next presenter is not online with us, so we'll go to the next presentation by Dr. Palanisamy Amritalingam from University of Tabuk, Saudi Arabia. And the title of the talk is Recent Updates in Pharmacovigilance. Hello, Dr. Palanisamy.
Hi.
Yes, so I'm going to make you the presenter. Yes.
Okay.
So please share your screen with us.
Is it visible?
Yeah, it is visible. You can make it a full screen.
Okay. Hello, everyone.
Yeah, can you please make it full screen? Please, yes, perfect. Continue with your talk. Thank you.
Yes. Hello, everyone. So I'm Dr. Palanisamy. So I'm here to present the topic on recent updates in pharmacovigilance in connection with the international webinar on pharmacology and toxicology with the theme of new technologies to identify adverse effects of chemicals or physical agents on living organisms. So in this regard, I would like to share my thoughts and views on the recent updates in pharmacovigilance. So this is my biography. So myself in Tabuk University in Coalesce as an assistant professor, and I completed my UG, PG, and PhD in Dr. MGR Medical University, Chennai, Tamil Nadu. And I'm teaching pharmacotherapeutics and pharmacy practice more than a decade. And my research interest includes diabetes, antibiotics, metabolic syndrome, etc. And I published numerous articles in peer-reviewed journals and participated as a speaker in national and international conferences. And currently an advisory board member for the Saudi Pharmaceutical Journal for Students, which is an official journal of University of Tabuk. So in this, so I would like to share the recent updates on pharmacovigilance and the prevalence and the pharmacovigilance cycle, and most importantly, the resources of pharmacovigilance system established by World Health Organization. And some educational point of view, the WHO established various free online programs regarding the pharmacovigilance. So that also I'm going to share with you in this presentation. So to start with pharmacovigilance. So what is pharmacovigilance? According to World Health Organization, pharmacovigilance is the science and activities related to the detection, assessment, understanding, and prevention of adverse drug effects or any other possible drug-related problems. As you all aware, the drugs. So all the drugs has their own corresponding adverse effects. And here the pharmacovigilance to understand, assess, and about to prevent such adverse effects to protect the human community from the hazardous effects of any drugs. So this is what the definition from the World Health Organization. So the pharmacovigilance cycle. So as you can see, this particular cycle established by Uppsala Monitoring Center. Uppsala Monitoring Center is a concern of World Health Organization, who is the headquarters for the pharmacovigilance system across the world. As you can see here, this particular cycle starts with the patient and end with the patient. So it starts with the patient. So after administration of a particular drug, if the patient experiences any noxious unintended drug reactions, the patient usually report or inform to the healthcare professional. So the healthcare professional, either physician, nurse, or pharmacist, usually recognize first about the particular adverse drug reaction. As I mentioned already, ADR is a noxious unintended response to any drug intended for the treatment, diagnosis, or any other purpose. So first of all, it is the ADR usually recognized by the healthcare professional. Then it is reported to the corresponding pharmacovigilance centers. Most of the countries across the world establishing their own pharmacovigilance centers. So the report will reach to the corresponding either regional or national pharmacovigilance center. So those centers usually collect and collate the reports, and those reports further analyzed, and the study will take place. So what is the purpose of this analyzing and study on the particular adverse drug reactions? As we already saw in the definition. So what is the importance of adverse drug reaction is prevention. So for that purpose, they used to establish the guidelines or therapeutic management in order to prevent or minimize the adverse drug reactions. So those guidelines usually communicated towards the healthcare professional. So that is all. The patient is the main stakeholder in the pharmacovigilance system. So this is what the pharmacovigilance cycle start with the patient and end with the patient. So here, as I mentioned already, WHO Uppsala Monitoring Center. So this is what WHO, Uppsala, Sweden. So who is responsible for collecting the adverse drug reaction reports across the world. And they used to make a database and they used to analyze, and accordingly the WHO make a suggestion or therapeutic guidelines for the corresponding adverse effects in order to improve in order to minimize or prevent the adverse effects. This is the prevalence. Why pharmacovigilance is very important. As you can see, the prevalence of area in worldwide. So this is for the actual for the calendar year for 2020. Unfortunately, I couldn't get for the last year, but still, as you can see in this pie diagram, antibiotic is predominantly contributing for adverse drug effects. Antibiotics are most widely used drug regimen in the hospital. So the antibiotics, again, it's responsible for contributing the majority of adverse effects, followed by anti-cancer drugs. 31% of anti-cancer drugs responsible for adverse effects, followed by anticoagulants, iron replacement, and most importantly, vaccines. So recently, COVID-19 vaccines have been established. Most of the vaccines initially uncertain. There is uncertainty for most of the vaccines. So everything were approved just for emergency purpose. So WHO, UMC closely watching these particular adverse effects from those particular vaccines, and followed by they used to come up with the guidelines in order to minimize the adverse effects with due to the vaccines. So this is what, and 20% of the remaining drugs has a single report at least in the calendar year 2020. So this is why it's important to recognize adverse drug reaction by the healthcare professional first of all, and most important thing is whatever did you recognize, you should report to the corresponding pharmacovigilance center. So this only makes to detect many adverse drug reactions, and those reports helpful to analyze and prepare for the therapeutic management for the corresponding adverse effects. So this is why the prevalence of ADR is keep increasing day to day. So that is why it is important to understand the pharmacovigilance system. The professionals, healthcare professionals and academia must understand and to report the ADR without any hesitation. In this regard, pharmacovigilance practice from WHO UMC, those who established 3 different resources, as you can see, World Health Organization, Uppsala Monitoring Center, establishing these 3 resources for pharmacovigilance practice: WikiFlow, WikiLice, and WikiBase. So here 3 different resources. So what do the WikiFlow does? WikiFlow is the strategical flow of pharmacovigilance system. As said already, the adverse drug effects usually recognized first by the healthcare professional. Then the report will go towards regional national pharmacovigilance center. Then finally, the report will reach to WHO UMC. So the WHO UMC will arrange the therapeutic guidelines in order to minimize or prevent such adverse effects through the database known as WikiBase. So WikiBase is a database which is maintained by WHO UMC, and WikiLice is about the signal detection. So what is the signal or signal detection? Signal is a quite new adverse effects. As you know, most of the drugs came across I think all of the drugs, not most, all of the drugs came across the various phases in the clinical trials, phase 1, 2, 3, and 4. Most of the adverse effects detected in these 4 phases. Unfortunately, some adverse effects usually detected even after the establishment of many years. For example, I could quote rofecoxib. Rofecoxib is the generic name in the brand of Viax. So that was established the manufacturer and very well accepted by the physicians as an analgesic. Unfortunately, the drug, after establishing quite few number of years, the drug was showing some cardiovascular complications. Then the drug was suddenly underwent for the pharmacovigilance, and they analyzed and they further banned that particular drug. And one more example, gatrifloxacin. Gatrifloxacin is one of the fluoroquinolone family member. That drug, that particular antibiotic, again very well accepted among the physician community. Unfortunately, the drug was causing hypoglycemia, and the drug has withdrawn from the market due to that reason. So everything, all of these things happens, it is due to the pharmacovigilance. And one more example I would like to quote, the pioglitazone. The pioglitazone is the PPR alpha sensitizer for the type 2 diabetes. So this pioglitazone, again, the diabetes management is one of the drug in the diabetic management in before. So what happens, suddenly there was many adverse effects on reports on the pioglitazone regarding the bladder cancer. Then the drug was withdrawn from the market. Again, the drug came once again into the market with the therapeutic management. How to monitor the drug closely on the patients like that. So, basis of drug use to drug causing such adverse effects being banned and came up again with a lot of therapeutic guidelines or therapeutic management or monitoring guidelines. It's all due to the pharmacovigilance practice. So here, 3 sources. So let us talk elaborately one by one. So as I mentioned already, VigiFlow is the strategic pharmacovigilance practice system. So here, 172 members belongs to the WHO program for international drug monitoring. So among these 172 members, 101 countries, those who are following complete pharmacovigilance system, are at least a part of such a system at year end. So what does it mean? The remaining 71 members, they are not at all following the WHO program for international drug monitoring. So it is important to understand what the WHO PIDM is doing, and the cooperation from the remaining country members is highly recommended by the WHO EMC. And we are in COVID-19 era, so EMC expanded in this regard VigiFlow in depth, going in depth, particularly the immunization programs, even at district, provincial, and national levels. And unfortunately, only 7 countries have been following in this particular the new system established by the EMC. Because as I mentioned already, there is lots of uncertainty. So most of the vaccines approved for the emergency purpose. So we are not sure about the vaccines, and there is no complete exclusion from any adverse effects. So that is why UMC went in depth in order to make a surveillance on the immunization programs. So this is about the flow. I mentioned it's the strategic pharmacovigilance system. Usually the report, the adverse effects usually reported by the healthcare professional, then healthcare professional usually recognize from the patient, and they forward to the corresponding pharmacovigilance centers. So here the countries has regional centers and the regulatory authority as a national pharmacovigilance center. So they used to forward these reports to the WHO VG base. So the countries, those who are following this flow, they are belongs to VG flow. So as I mentioned already, here 101 member countries, those who are following this particular system. Patient to the healthcare professional to the National Pharmacovigilance Center, then the National Pharmacovigilance Center towards World Health Organization (UMC). So this is what the flow usually maintained most of the member countries of WHO UMC. And this is the other resource. It's a database, as I mentioned already. It's a unique database regarding the report of potential side effects of medicinal products and continuously updated. And here almost 27 million individual case safety reports have been received so far by the WHO PIDM program since 1968. As you can see in this graph, it has established from 1968. Now, towards 2022, it is reaching almost 27 million. And most importantly, as you can see here, 4 million reports, 4 million individual case safety reports from the low middle income countries. So this is what the strength nowadays is supporting WHO EMC like anything. So hopefully the WHO EMC will elaborate their vigilance system across all over the world thoroughly in order to improve the human health. And this one is the VG base report, according to the annual report of WHO EMC. As you can see, the map, world map. So this one is ICRs, the individual case safety reports per million inhabitants. So here, the picture, as you can see in the dark gray, so those who are reporting more than 500 per million, more than 500 reports per million population. So ADR reporting is quite high, as you can see, starting from more than 500, and very light gray, the countries, those who are just reporting less than 5 reports per million inhabitants. So recent report of WHO UMC, the pie diagram which shows the country distribution for individual case safety reports for the 12 months for the last year. As you can see, USA and the remaining countries are contributing towards drug reaction report WMC. And these are the members. These are the members. As you can see, the highlighted blue, dark blue, those were the full members of the WHO PIDM program. And in blue, in sky blue, it is about the associate member. And circled in brown, those were the very recent members of WHO PIDM program. So this is what the good news. Most of the countries are participating in WHO EMC. However, but still countries in Africa and other regions of the world are still yet to participate in WHO PIDM program. And the frequency of ICSR reporting, this is time since last submission. The first, the left-sided pie diagram which shows, and 66.2% of member countries, those who have reported for quarter, and 8% reporting only those who are 3 to 6 months, and still did not report any adverse 16.2% more than 12 months. And the other pie diagram in the right side, which shows the number of countries submitting ICSRS in Wikibase, and 88 member countries, those who are reporting almost all the quarters towards WHO, and only the 30 countries shared over the past reports. So this is the thing. International community is particularly international conference on the countries those who are representing vigilance reporting in order to improve and protect human health. And the other one, the Vigilize, is the other resource. As I mentioned already, it's about the signal detection. The signal is a quite new area which was not previously recognized by any healthcare professional, even after the post-marketing surveillance. That could be called as signal detection, and that uses insights into the safer and use of medicines from the members of WHO PIDM, and as a starting point for efficient quantitative signal detection. The signal is the first suspicion of drug area arises. So who will recognize? The healthcare professional. And the signal, that particular new adverse drug effects, strengthened by the data, various data and value of cases, and proportionately data from the other resources, literature and case series, etc. So finally, the Vigilize usually gain more advanced statistical signal analytical capacity, and vaccine by vaccine, drug by drug, and as well as the analysis across the different vaccine or drug platforms. And in this regard, Vigilize updating their data twice a week in order to evaluate the corresponding signals detected by the healthcare professional reported across the world. So this Vigilize resource will be helpful to understand about the signal detection. So this one is the Wiki Access. So Wiki Access, this is the other platform offered by WHO PID Monitoring Center, which was established in the year of 2015, which consists the adverse effects of all the drugs which was already reported to WHO UMC. If you go for the access of wikiaccess.org, then you can find the search column. So if you find the adverse effects of the corresponding drug, just type the drug name. Methotrexate have been typed by me, and I'm looking for the adverse drug reactions of methotrexate. So these are the adverse drug reaction reports have been to WHO UMC in the system wise, starting from blood, lymphatic system, cardiac disorders, etc. Here I went through skin subcutaneous tissue disorders. See, almost 17,000, almost 18,000 reports regarding skin and subcutaneous tissue disorder alone due to methotrexate. And if you go for further categorization, alopecia, 4,000 reports regarding alopecia, and here I'm going to erythema multiforme. So 76 reports across the world. So if you want to find any adverse drug reaction reports in WHO UMC database, you can very well access this Wiki Access platform. And WHO develops and spreads knowledge on scientific methods, best practices, and provides guidance on the use of data management and analysis tools. This context, WHO offers various free online courses for the healthcare professional and the students. So that I'm going to discuss in the later slides. So before that, and they developed scale which was organized to a so scale which was known as WHO UMC causality assessment scale. So what is causality? Nothing but finding a causal association or relationship between the drug and drug reaction. So is there any relationship between the drug and drug reactions? So we need to make sure. We need to make what is the level of relationship. Here 4 different level of relationship between drug and drug reaction: certain, probable, possible, and unlikely. So
Hello, Dr. Palanisamy.
or other
Yes, your voice got disrupted. Hello.
now?
No, it got stuck somewhere.
I'm sorry. I'm sorry for that.
Yeah, now you can continue from this slide, WHO-UMC. Again, it got disconnected, your audio.
Really?
Yeah, now it is okay.
Hello?
Please continue. Yes, now you can continue.
Okay. So, these are the criteria and the recovery after withdrawal and the recurrence after re-challenge. So here, any adverse effects we use to withdraw the drug. So after withdrawing, is there any recovery or not? And after re-challenge is introduction of drugs for the already which was detected with the particular adverse drug effect. And re-challenge is again reintroducing with the low dose. So if S for all these criteria, this is known as certain. That means the relationship between drug and drug reaction is very strong. And probable in this case, yes to first three criteria. However, no or there is no data regarding the re-challenge. In that case, it is the probable level of the relationship. When it is possible, just time relationship, the administration of the drug and the appearance of adverse drug reactions. So is there any association? Only yes. So it is possible relationship. And unlikely, no to all criteria, it is known as unlikely. So here the one important thing is re-challenge is usually not recommended for the serious adverse drug reactions. And unlikely, in case of unlikely, it is suggestive if event resolves despite the continued exposure. So this is regarding unlikely. For example, if the drug causing certain adverse effects, however, the adverse effects is usually resolved over a period of time of continuous administration, that could be considered as an unlikely. So this scale will help the community, the healthcare community, to assess the relationship between the drug and the adverse drug effects. The same should be reported to the Pharmacovigilance Center for the further analysis. So as I mentioned already, there was various free online courses offered by WHO UMC. So this is the link for the particular courses, and you can simply go in the Google search engine and you can search whoumc.org. You can find the pharmacovigilance trainings and go and click the e-learning platform. So here was many free online courses offered by WHO UMC. So this is free for the healthcare professionals, those who are practicing in the hospital, pharmacy, and academia. So this is free for the academic the professionals also. And here the courses are available in English and Spanish, etc. So these are the 8 different courses regarding the pharmacovigilance. As you can see here, introduction to pharmacovigilance and essential to understand the basics of pharmacovigilance and collecting high quality ADR reports. So this is the other course, and as I told you about the causality assessment. So how to do the causality assessment in a comprehensive manner, they will teach you, the WHO will teach you. And the remaining 3 courses, as you can see in the bottom, about the signal detection, introduction, signal assessment, and statistical reasoning for the signal evaluation. So all these 8 courses for free for the healthcare professionals and even for the students and academic professionals. So I call upon all the respected delegates, those who are joined in this particular platform. So try to convey this message with your colleagues and maybe useful for yourself also. And the people, those who are looking for the job opportunity in pharmacovigilance centers, this will be very helpful for them. And here I would like to conclude with the take-home message. The pandemic has made the global pharmacovigilance security more important than ever. So this was this tough situation. It is important to join hands together to succeed against the COVID-19 by making certain therapeutic managements by ruling out the adverse effects, both the drug treatment and as well as the immunization programs. Alongside the efforts of the medical and scientific community to find suitable treatments and vaccines for COVID-19, as I mentioned, and the WHO has also been vital to the success of public health initiatives across the globe. So, I would like to conclude with my sessions. The pharmacovigilance is a very important area, as my previous presenter, as he mentioned, particularly in this COVID-19 issues, and not only for the natural products. It's very prominent to practice pharmacovigilance practice in the modern medicines and vaccines, etc., in order to improve the global health. So this is my reference annual report of WHO EMC. This was the recent report released by WHO EMC. And thanks for your attendance. And let me hear from you if you have any questions. And before I conclude, I would like to thank the organizers of this particular international webinar. Most fruitful information for the past 2 days across the scientific community across the world. And, thank you so much. Thank you so much for everyone. So let me hear from you if you have any questions.
Thank you, Dr. Palaniswamy. It was nice hearing from you. So is there any questions from anyone? Okay, if you have any questions also, you can mail us. We will forward it to Dr. Palaniswamy and will answer you the questions. So thank you once again for your presence.
Thank you so much. Thank you so much.
Yes.
Thank you.
Thank
Have
you.
a nice day. Thank you.
Have a nice day. So our next presenter is Dr. Amir Mohammad Bezai from Maznadaran University of Medical Sciences, Iran. And the title of the talk is Comparative Efficacy of Fractional CO2 Laser and Q-switched ND-YAG laser in combination therapy with tranexamic acid in refractory melasma, results of prospective clinical trial. So hello, Dr. Amir.
Hi.
Yes.
Welcome.
So, yeah, hi. So you are the presenter now. Please share your screen with us.
Yes. Only one moment. Okay.
Yes. So you are going to present through mobile device.
Yes, it's by my cell phone.
Okay.
Is it okay?
Yes, yes, you can.
Okay.
But it is hard to visible the content. Can you please zoom in somewhat, like so that we can able to see the content?
Yes. Is it okay right now?
Yeah, or else it will be possible for you to make it horizontal. Will it be possible?
Mm. Is it better now?
Yeah, just a minute. Yes, yes, now it is perfect. Yes.
I made it horizontal, I think.
Yes, please continue.
Thank you. Welcome and greetings. Thank you. Welcome and greetings to the conference. Good morning, dear professors and colleagues. Hi, let me briefly introduce myself. I am Dr. Beyzai, graduated from Mazandaran University of Medical Sciences. Thank you for taking your precious time. I'm delighted to be here today to talk to you about a new promising combination therapy for refractory melasma, which is still challenging to both physicians and the patient. Let me start with some general information about melasma. Melasma, an acquired perivalent pigmented disorder, manifests as hyperpigmented macules and patches. The prevalence of melasma is higher in young women with 4 to 5 Fitzpatrick skin type. A positive family history, UV light exposure, and hormonal factors are the most common risk factors associated with melasma, as evidenced by flare during pregnancy and use of oral contraceptive pills. Treatment modalities for melasma can be broadly divided into medical and cosmetic therapy. Medical therapy including oral and topical medication, and cosmetic therapy is consisted of laser therapy, phototherapy, and chemical pills. Topical hydroquinone and the combination of hydroquinone, tretinoin, and fluocinolone acetonide, which is known as a triple therapy, are still considered as the gold standard treatment for melasma. Also, tranexamic acid, which is an antifibrinolytic drug, is found to be effective both in its oral as well as topical form. Various lasers and light sources have been proposed in the treatment of melasma. Q-switch Nd:YAG laser, fractional laser, and intense pulse light are energy-based treatment modalities used in the treatment of hyperpigmented skin lesions. Fractional carbon dioxide laser produces dermal microdermal zones and it is used in the treatment of scar, but also superficial pigmentation such as melasma. Despite available options, treatment of melasma can be challenging. Tranexamic acid is an analog of the lysine amino acid, which inhibits plasmin activity by binding to the plasminogen in the keratinocytes. In addition, through mechanisms such as decreasing tyrosinase activity in melanocytes, increasing vascular endothelial growth factor or VEGF, and alpha melanocyte stimulating hormone, it can be used as a treatment modality for melasma patients. On the other hand, laser therapy facilitates the penetration of tranexamic acid into the affected tissue. Therefore, a lower dosage of tranexamic acid is required to achieve the desirable therapy goal. In this regard, we decided to conduct a comparative study to evaluate the efficacy and the safety of a fractional CO2 laser with a Q-switched Nd:YAG laser in combination with tranexamic acid in refractory melasma. In this study, 30 patients with refractory melasma were enrolled. Refractory melasma refers to the patients who have been treated at least with 2 standard medications for a minimum duration of 3 months and had less than 25 improvement, according to the modified Massieu score. The fractional CO2 laser with power 30 watt, pulse energy 30 millijoule, tip type 300, pulse rate 100 per meter per square centimeter. ECO2 device Lotronic company made by Korea was applied to one side of the face and 3 passes of the Q switch NDIAG laser with the wavelength 1064 nanometer, pulse energy 750 millijoule, fluence 1.5 joule per square centimeter, spot size 4 in 4 millimeters, and piece fractional Jesus company made by Korea were applied to the other side. A total of 6 sessions of laser therapy was given with 2 weeks interval. Throughout the 12 weeks course of laser therapy, all patients received 250 mg of oral tranexamic acid twice a day. Treatment efficacy was evaluated using the patient's global assessment and the physician's global assessment scales. Objective evaluation was based on improving the intensity of skin pigmentation by viewing serial images taken from hyperpigmented lesions. In the initial assessment, before the start of the process, the patient's facial hyperpigmented lesions were photographed by a digital camera. Then, in the follow-up sessions, 4th, 8th, and 12th weeks, the extent and amount of pigmentation was compared to the first session, and the mentioned indexes were re-evaluated. After treatment, patients were followed up for another 12 weeks. Adverse events were recorded. The treatment satisfaction questionnaire for medication, TSQM, was used to assess the treatment satisfaction. The study was approved by the Institutional Ethics Committee. Written consent was obtained from all patients. The exclusion criteria was consisted of patients with autoimmune diseases, hepatitis, coagulopathies, and thyroid disorders. The patients who have been treated with phenytoin, tranexamic acid over the past 1 month, retinoid drugs over the past 3 weeks, topical medication to improve skin pigmentation over the past 1 month, hormone therapy such as OCP, anticoagulant agents, or laser therapy to treat melasma over the past 6 months. A positive history of allergy to tranexamic acid was excluded. Pregnant women or breastfeeding mothers were excluded. Patients expressing dissatisfaction with mentioned treatment techniques were excluded from the study. A total of 30 female patients with a mean age of 39.97 years, with a minimum age of 28 years, and a maximum age of 66 years were included in this study. The mean duration of disease was 6.55. Patient global assessment in fractional CO2 laser group was significantly better than the Q-switch NDIAG laser group at the 4th, 8th, and 12th weeks. There was not a significant difference of patient satisfaction between the groups. As you can see in the table, the difference between the patient global assessment at the 4th, 8th, and 12th weeks is significantly different between the two groups, and in fractional CO2 laser is better. As you can see, the p-value is less than 5%. But patient satisfaction, the difference between patient satisfaction between the two groups is not significantly different. Patient global assessment improved significantly in both groups over time. There was an interaction between time and the groups, meaning that patient global assessment in the fractional CO2 laser group improved more than the Q-switch NDIAG laser over the time, and this difference was statistically significant. Physician global assessment at the 8th and 12th weeks and physician satisfaction at the 8th week in the fractional CO2 laser group was significantly better than the Q-switch NDR laser group. As you can see in this table, at the 8th and 12th weeks, the difference between physician global assessment is meaningful, as the p-value is less than 5%, and in fractional CO2 laser group is better than the Q-switch NDR laser. Also, in the physician satisfaction and 8th group, at the fractional CO2 laser is significantly better than the Q-switch NDR laser, according to the p-value. The physician global assessment in both groups significantly improved over time. There was no significant difference in the rate of physician global assessment reduction between groups over the time. The Massieu score in the fractional CO2 laser group was significantly lower compared to the Q-switch NDR laser group at the 8th week. The Massieu score significantly decreased in both groups over the time. The Massieu score in the fractional CO2 laser group decreased more than the Q-switch NDR laser over the time. As you can see here, at the 8th week, the difference between Massieu score in the both groups is significant, according to the p-value, which is less than 5%. The most common reported side effects were erythema and discomfort, which subsided in less than 24 hours. There was no significant difference in the frequency and type of adverse events between the groups. In conclusion, the fractional CO2 laser was found to be superior to the Q-switch NDR laser. The results of our study show that a fractional CO2 laser with oral tranexamic acid is an effective and well-tolerated therapeutic method for the treatment of patients with refractory melasma. Thank you for your precious time. If there is any question, I'll be happy to answer.
Thank you, Dr. Ahmed, for your presentation. So any questions from anyone? So no questions. Thank you once again for your presence and presentation. So if anyone have any questions, you can mail us. We will forward it to Dr. Ahmed.
Thank you for your great webinar and conference. Thank you very much.
Thank you very much. So as we can see, our next presenters are not there and they will join after some times as we have contacted them. So we'll go to the poster presentations now. So can I request Dr. Lakshmi Singh, can you able to present the presentation now? Hello, Dr. Lakshmi. Hello.
Hello.
Yes, Dr. Lakshmi, can you able to give your presentation now?
Yeah, ma'am. I'm ready.
Okay, so I'm going to make you the presenter. So next is poster presentation presented by Dr. Lakshmi Singh from Dr. B.R. Ambedkar University, India. And the title of the talk is Placental Concentrations of Metals and Their Association with Preterm Delivery, Indian Scenario. So you are the presenter now. Please share your screen with us.
Sure, ma'am.
So is there any issue, doctor?
Yeah, ma'am, I'm trying to share my screen, but
Yes, please click on the screen icon. Is it a MacBook?
yes, ma'am, I am using MacBook.
And you need to give the extra permissions in system preferences. So once you give that one, once you log out from the session and again try to join,
Open
and then you
system
can able.
preference, ma'am.
Yes, so you're there, you need to give the permission to share your screen with others. So did you give the permission?
Yes, ma'am, but
Okay, then what you can do is once you log out from the session, once you leave the session and again try to join.
okay, ma'am,
Yes.
I'm trying to.
Yes, so you can once leave the session. Can we proceed with another talk? In the meanwhile, you can check with your settings, and I will suggest you that once you close the webinar and try to join again, and then it will definitely work and you can able to share your screen with us.
Okay, ma'am, I tried.
Okay. Yeah, you can leave the session and again try to join from the link as same link. And in the meanwhile, we'll proceed with another talk. So if you have any queries also, you can ask me. I can guide you through chat process. Yes. So as Dr. Amir has arrived the webinar. Hello, Dr. Amir. Can you able to give the presentation now?
Hello, madam. Yes, I can. Yes, I can.
Okay. I'm going to make you the presenter.
Okay.
Yes. So our next talk is from Dr. Amir Sharam from Sfax University, Tunisia. And the title of the talk is Effects of Caffeine Ingestion on Psychomotor State and Oxidative Stress Markers after an 8 km run competition in sleep-deprived recreational runners. So Dr. Amir, you can click on the screen icon. Yes, we can able to see your screen.
Okay.
Yeah.
Is it clear?
Yes, perfect. Please continue with your presentation.
Okay. Hello, everybody. My name is Amir Sherm from Tunisia. My communication is entitled Effects of Caffeine Ingestion on Psychomotor State and Oxidative Stress Markers after an 8-kilometer run competition in sleep-deprived recreational runners. It was directed by Professor Zuhair Sahnoun. In this study, we have 4 sections. We begin with the introduction and problematics. Second, we will see the material and methods. After that, we will see the results and discussions, and finally, the conclusions. We begin with the introduction. We will see the main parameters of the study. The first parameter is caffeine. As we know, caffeine is the most consumed psychoactive substance in the world. We could find caffeine in the coffee. We could find caffeine in the tea and chocolate and soda. Also, caffeine is a powerful central nervous system stimulant. And caffeine could lead to improved alertness, strength, and endurance. Also, caffeine could lead to improved sports performance. The second parameter of the study is sleep. As we know, we have 4 kinds of sleep. We have very light sleep, deep sleep, and paradoxal sleep. Sleep is a process of repair and homeostatic regulation. It is also the adequate sleep is from 6 to 8 hours per night, and it is fundamental for health. However, sleep deprivation could alter physical, mental, and cognitive state, and these effects are more pronounced with total sleep deprivation, TSD, that is exceeding 24 hours of sleep deprivation. The third parameter is oxidative stress. What is oxidative stress? It is the main reason for the deterioration of cells. It is also an imbalance between the production of free radicals, FR, and the antioxidant capacities of the body. However, the defense system of our body have enzymatic antioxidants such as SOD and GPX, etc. And we have also non-enzymatic antioxidants such as GSH and uric acid, etc. And this defense system will lead to the trapping or the inhibition of free radicals production. Example in long and aerobic exercises. For the problematic of the study, although caffeine is a commonly used aid in athletic performance, especially among top athletes, few arguments exist to justify its restorative effect on psychocognitive parameters and those of oxidative stress after total sleep deprivation, TSD. We have 2 questions. The first question is, could caffeine supplementation prevent the deleterious effect of total sleep deprivation following aerobic exercise? The second question is, does caffeine exert an antioxidant effect during this type of exercise? The second section now is material and methods. We begin with the objectives, then we will see the participants, after that the protocols, and finally the tests. The aim of this study is to analyze the effect of the ingestion of 5 milligrams of caffeine 1 hour before the race of 8 kilometers, after a reference night, and also after a total sleep deprivation night on 4 parameters: on the physical parameter, on mood, on cognition, and also on oxidative stress parameter. About the participants, we have 10 participants. Their age is about 21, mass about 66, height about 1 meter 0.76, body mass index about 21, and VO2max is about 51. For the protocol of the study, we have 4 test sessions. We have 2 test sessions after a reference night, RN, and 2 test sessions after total sleep deprivation night, TSD. Following in the reference night, RN, the participants will sleep from 22:30 to 7. And for the total sleep deprivation night, participants will not sleep. After that, at 7:30, subjects will ingest 5 milligrams of caffeine in capsules. Following that, at 8:30, we will realize the blood intake and the psychomotor tests. After that, at 9 hours, participants will begin the running of the 8-kilometer competition. And in the end of exercise, we will repeat the blood intake and the psychomotor tests. Here we will see the tests realized in this study. First test is the 8-kilometer running. It was realized around a 400-meter outdoor athletic track. It is the track of the University of Sfax, Tunisia. Then before and after the running, we have the psychological tests. We have 2 psychological tests. We have the feeling scale test and the Hooper test. In the feeling scale tests, FS, participants will evaluate the feeling before and after the running, from the very bad feeling to the very good feeling. And for the Hooper test, participants will evaluate the sleep, the stress, the fatigue, and also the pain before and after the running of the 8 kilometers. Following that, we have the cognitive tests. Also, we have the 2 tests. We have digit cancellation test and reaction time tests. On the left here, the digit cancellation test. It consists of the cancellation of all numbers with 3 digits in 1 minute. And for the reaction time on the right, participants will push the space bar of the computer as quickly as possible for 10 times. And finally, we have the collection and the treatment of blood samples before running and after running. The collection of blood samples, as we see here, it will be realized just after the running. Then the blood samples will be taken in the laboratory for the analysis. And finally, now we'll see the section of the results and the discussions. For this study, as we said before, we will see the results in the physical parameters, the mood, the cognition, and the oxidative stress parameters. For the physical parameter, we will see the effect of caffeine on the physical performance, which is the performance of the 8-kilometer running. As we see in this figure, we have in blue the placebo group and in red the caffeine group. In the left, we have after the reference night, and in the right, we have after the total sleep deprivation night. As we see in this figure, the times recorded for the caffeine group are less than the placebo group, which is best than the placebo group. That is, the caffeine group is best following the reference night and also following the total sleep deprivation night. So the administration of 5 milligrams of caffeine significantly improved running performance after reference night as well as after total sleep deprivation night compared to the placebo group. Also, we could see in this figure that there is no significant difference between the performance of the control group after the reference night and that of the supplemented group after the total sleep deprivation night. Thus, caffeine seems to restore the physical performance, which is affected by the total sleep deprivation. This recovery could be due to the antagonistic effect of caffeine on adenosine receptors, which could improve wakefulness and reduce drowsiness. Now we see the psychological state. We have two tests. For the test of feeling scale, as we see here, in red, the caffeine group, and in blue, the placebo group. As we see here, the caffeine group is usually best than the placebo group in the reference night and also after the total sleep deprivation. So the consumption of 5 milligrams of caffeine increased the feeling of well-being after running competition following the reference night and total sleep deprivation. Thus, caffeine could be effective in minimizing negative psychological effects induced by sleep loss conditions. Now we pass to the second psychological test, which is the test of Hooper. We have 4 parameters: stress, pain, fatigue, and sleep. As we see in this table, the stress was reduced from 4 to 3.5 after the administration of caffeine. Also, for the fatigue, it was reduced from 6.3 to 5.4 after the consumption of caffeine. We could say that caffeine consumption after total sleep deprivation reduced feelings of stress by 18.3% and feeling of fatigue by 14.3%. So the analgesic properties of caffeine seem to help delay fatigue after reference night or total sleep deprivation, which improves psychomotor state and physical performance. Now we pass to the cognitive state. We have also 2 tests for the reaction time test, and we have the test of digit constellation of numbers. For the reaction time here, we could see in this figure that caffeine have best times after reference night, and also it has best times after total sleep deprivation night. For the digit constellation of the numbers, we could see that the caffeine group have more numbers than the placebo group following the reference night and also following the total sleep deprivation night. So caffeine consumption led to faster reaction time and better attention after reference night and total sleep deprivation. This beneficial effect of caffeine on the psychic state could positively affect the cognitive states of athletes despite their alteration following TSD. Finally, the oxidative stress parameter. We have 4 parameters. We have GPX, GSH, and SOD antioxidants, and we have the peroxidation parameter of MDR. We could see here in this table that there is no effect of caffeine. We have only the effect of the sleep or the exercise. For example, the GPX was reduced from 8.4 to 6.7 following sleeping. However, the MDR was elevated from 5.2 to 6.1 following the exercise, and there is no effect of caffeine. So caffeine does not appear to improve oxidative stress status after reference night and total sleep deprivation night, and the observed changes were influenced only by exercise and sleep condition. In this way, current experimental evidence in animals suggests that sleep deprivation promotes oxidative stress by lowering antioxidant defenses. In the conclusion of this study, we could say that the administration of 5 milligrams of caffeine 1 hour before the running of 8 kilometers following a reference night or a total sleep deprivation night could lead to improved performance also could improve mood and cognition without affecting the oxidative stress parameters. This study led to publish 2 papers in the Biological Rhythm Research Journal. And finally, thank you for your attention.
Thank you, Dr. Amir, for your presentation. So any questions? No questions. Thank you once again for your presence and presentation.
Thank you for this opportunity. Thank you.
Thank you. So we'll go with the Dr. Lakshmi Singh presentation. Hello, Dr. Lakshmi. Hello. Hello, Dr. Lakshmi. Am I audible to you?
Hello.
Yes. So you are the presenter now. Can you please once try with sharing your
Yes.
screen?
Yes, ma'am. I'm trying it, but
Yes.
still facing same problem, ma'am.
Okay. Okay. So what we can do
Open
is we have your.
system preference is not allowing.
Okay, no issue. We have your poster presentation. We will share our screen. You can give the explanation from there. Is it okay?
Okay, ma'am.
Okay, then we'll be sharing our screen. Just allow me a 1 moment. Can you able to see our screen, Dr. Lakshmi?
Yes, ma'am.
Yes, so you can start your presentation. If you want to scroll down or up, you can just let us know. We'll do that for you.
Okay, ma'am.
Okay.
Hello, everyone. Good afternoon. Firstly, I would like to thanks all committee members of Toxicology and Pharmacology for giving this opportunity for me. Today, I am going to present my research work entitled Placental Concentration of Matters and their association with preterm delivery in Indian scenario. As we know, pregnancy is an important and most beautiful stage of each and every woman's life. And it is generally observed that healthy pregnancy lead to the strong foundation of the healthy life of the infants. But during this period, childbearing population are exposed by number of unwanted pollutants, and these all pollutants are responsible for adverse pregnancy outcomes like preterm and low birth weight. According to World Health Organization, preterm birth may be defined as if any child take birth before 37 weeks of gestational age, and it is called preterm birth. Preterm delivery remains the leading cause of prenatal mortality, and it occurs approximately in 7 to 11% of the pregnancies. According to a recent report, more than 50 million babies are born preterm, and these approximately more than 1 million preterm babies are die annually in developing countries like India. According to World Health Organization, India is the biggest contributor to the world in case of the prematurity burden, and approximately 85% of premature babies are low birth weight. In northern India, especially in case of Agra region, preterm birth and low birth weight are two major causes of neonatal mortality. As we can see in this figure, India is the biggest contributor in prematurity cases. There are the number of factors responsible for preterm deliveries, and these factors are divided into two categories. First is modifiable, and second is non-modifiable factors. Non-modifiable factors are not improvable during pregnancy and have not direct influence on the pregnancy population. But in case of the modifiable factors, these factors are improvable during pregnancy and have direct influence on the pregnancy population. For example, smoking, addiction, and environmental pollutants like metals. These metals are present in everywhere in an environment, and easily accumulate in a biological tissue because they have probability easily bioaccumulate in a tissues. And apart from this, some metals are easily transfer mother to fetus with help of placental, and they act as a endocrine disruptors in placenta. And some metals are required for body, and these metals act as a enzymatic play important role in enzymatic activities. But some metals are not required by body, and such type of metals are highly toxic at a low concentrations. Keeping these all points in a mind, the objective of my research work are to compare metal concentration in placental tissue of the preterm deliveries and with normal deliveries, that is full term delivery. And additionally, I have tried to find out the levels of lipid peroxidation, that is MDA, and antioxidant GSH in both cases. And we have tried to find out the correlation between redox status and some methods. For this work, firstly, I have collected approximately 100. I have collected 150 placenta from healthy pregnancies, and I did collaboration with SN Medical College in Agra. And additionally, data were collected by personally interview from participants after delivery for know about socio-demographic and lifestyles of each enrolled females in this study. Now we comes on the most important points, the finding of my study. My first table represents socio-demographic characteristics of the enrolled women in this study. In this table, no significant difference were observed in maternal age, height, weight, and number of children, and BMI between two groups, except gestational age and hemoglobin. And the first figure of this poster represents GSH and MDA level redox status in a placenta. The finding of present study shows that GSH content was significantly greater in the full-term deliveries compared to preterm delivery, whereas MDA content was significantly higher in preterm delivery group compared to full-term delivery group. And the increased placental MDA concentration and lower concentration of GSH in a preterm patients compared to full-term cases revealed that GSH defense were directly utilized in detoxifying the excess free radical in the placenta. And second table of this poster represents metals concentration in both groups, preterm and full term. In this table, cadmium cadmium, arsenic, and lead were significantly higher in preterm group compared to full-term group, whereas zinc and iron were significantly higher in full-term group than preterm. Additionally, we observed that nickel and aluminium were higher in preterm, whereas the difference were not significant. And now we comes on the second figure of this poster. Please scroll down. Yeah. This figure represents in figures cadmium and lead have significant positive correlation with MDA, as we can see. And these figures provoked oxidative stress and suggest increased oxidation stress leading to increased rate of the lipid peroxidation in preterm deliveries, which may be contributed in prematurity and its complication. Whereas negative correlation between placental lead and GSH, indicating lead-induced disruption of oxidant and antioxidant balance in a placenta in pregnant women. And second table of this poster tells us about the potential source of the metals, and for find out the potential sources of metals, we have analyzed principal component analysis and find out three main sources of the metal emission in the environment. First is combustion of biomass fuels. Second, water. Second, dietary source. And third is water contamination. And now last, we comes on the conclusion of this work. As we have seen, the toxic metal were higher in my study. These results indicate that levels of essential and toxic metals is related to unnecessary elevated levels of oxidative stress, and it diminished the protective mechanism such as GSH could be part of the preterm deliveries. Significantly, levels of cadmium and lead and arsenic, aluminium, these all levels are harmful for preterm for pregnancies, and significantly elevated levels of lead and cadmium were significantly correlated with MDA, suggest toxic metal induced preterm deliveries in women of the present study. Though the altered oxidants and antioxidant status in a placental tissue. And finally, according to PCA analysis, the sources of exposure to the study population were biomass fuel combustion for food habit and drinking water are the main source of exposure in pregnant women. Thank you. Hello. Hello.
Hello. Yes, thank you, Dr.
Hello.
Yes. Hello. Am I
My
audible
voice is
to you?
audible?
Yes, yes, it is audible.
Okay, ma'am. Any queries?
Yeah, thank you, Dr. Lakshmi, for your presentation. So, any questions from anyone?
It's me, Dr. Gulo.
Yeah, hello, Dr. Gunda.
Just
Yes,
to
continue.
congratulate with the colleague. I want just to congratulate with the colleague for the really excellent work, because I'm a gynecologist, as you can see, and really interesting about the study about placenta, and that's all. Congratulations.
Thank you, sir. Thank you, ma'am.
You're welcome.
So, is there any else questions? No questions. Thank you once again for your presence and presentation.
Thank you, ma'am.
Yes.
for
So
giving
as
this
we
opportunity.
thank you very much. So as we can see that Dr. Sumaya Zareen Sheikh has arrived the webinar. So our next presentation will be from her, and she is from the SIA College of Health Sciences College of Physiotherapy, India. And the title of the talk is Error and Biases in Research Methodology. Hello, Dr. Sumaya.
Hello, good afternoon or good morning towards the British side. Can I share my screen?
Yes, you are the presenter now. Please share your screen.
Okay.
Yes, you can. We can able to see your presentation. No, or screen. You can open the presentation.
Yeah, can you see now?
Perfect. You can make it a full screen.
Okay, yes. Should I
Yeah,
start?
perfect. Yes, you can start.
Okay. Hello, everyone. The title of my topic today is Errors and Biases in Research Methodology. We know that research, the basic principles of research are two, or you can say the two pillars of research are transparency and accuracy. However, just 1 minute. Yes. So, however robust is your methodology, or however you want to, communicate via scientific writing to the audience, you have to. We all know that the stakeholders are the authors, the readers, and the reviewers. Now, all three of them maintain transparency and accuracy of your research or your communication, which you're going to do via scientific writing. But the initial responsibility lies on the authors. And reviewers ensure that it is up to the mark, it is transparent and accurate. And readers also ensure these two basic principles of research are maintained by reading your content, and maybe if they find some loopholes or unstrategic planning, they can always write letter to editors, to the journals where it is published. So the initial and the main responsibility is of the authors. Now it is our understanding how to make this methodology very robust. So how you design, how you conduct, and how you report. These are very simple terms to say, but actually there are small things where sometimes we don't understand what are the reasons for rejection, because reviewers can easily pick these things up. So it is a very wise decision sometimes to introduce, or always to introduce, a statistician in the beginning, and do not introduce a statistician at the end after you have reported, conducted, and designed your methodology. Because once the biases or the errors, if they are not clearly fulfilled in the methodology, you cannot go to a statistician saying cover up for all these biases. Instead, we design a robust methodology. So the objectives of today's lecture are what are the guidelines for especially designing observational studies, because we are more focused on reading RCTs. We are more focused on reading case control studies. But what about observational studies which are more feasible, which do not, have a funding backup? And these are more of the studies that we usually do. So not all studies can have a funding, a good database, and a good feasible methodology. So let us see what are the errors and what are the biases and what sources of biases and the types of biases. Certain times after, when we are done with making a manuscript, we usually go for guidelines or checklists. And these are the checklists over here for I'm going to talk today only about observational or cross-sectional study designs. And we usually look for these studied checklists after our methodology is designed or after our manuscript is ready. So it is better that while designing a study design, or while when you're thinking of preparing a study, why not make it as a prerequisite prerequisite for designing these checklists. like strobe, Pedro, Robin's eye, and the NIH tool. So let us see strobe first. Strobe is the strengthening of the reporting of observational studies in epidemiology. You can see it is actually a 22 item checklist. Only 21 are present here. But this is especially meant for designing observational cross-sectional case control and cohort studies. And it contributes to the methodological conduct of the design, and all authors, editors, reviewers, they all use strobe to actually critically appraise any study. So instead, when you know that your study is going to be analyzed using these guidelines, why not introduce it in the methodology? You can see how the title, introduction, variables, methods, results, participants, the other analysis all have been given an item number, and they are expected to be answered by the author in the manuscript. Okay, and if you have any difficulty in understanding what are they expecting in the study design, the explanation is given here. We move forward to Pedro. Pedro is especially for physiotherapy studies. It is almost like strobe. It is a small 11 item checklist. But why I have included this over here, it was actually initially funded by the Sydney University and the APA. And you can also do courses for Pedro. And if you do those courses for Pedro, you are known as friends of Pedro. So it's a simple 11 item checklist where you have to just answer these questions. For example, let's take the question number 5. Was there blinding of all subjects? Question number 6 is, was there blinding of all therapists or maybe the administrators who administered the therapy? Yes and no. If it is no, or if it is yes, where in the manuscript? So usually you can, line number your manuscript. You can write here where exactly on maybe line number 239 did you see that the statement regarding blinding was written. So it is a good habit to do it before you submit, and actually when you're designing a study design. So you have all these 11 similar questions. Moving to Robin's eye. Robin's eye is especially used when a meta-analysis is done. So to assess the risk of bias, first of all, it is important by using these two guidelines, Strobe and Pedro, we will understand or we will minimize the sources of biases. Okay, but biases are going to enter into your study by hook or by crook. So how do you analyze how much is the risk? So the risk of bias, this is a study by Stern and actually Cochrane, published in 2016 by the BMJ. So especially for observational studies, because the methodology is different, Robin's eye or observational studies are also called as non-randomized controlled trials. So they are non-RCTs. And they not only identify biases, but they also calculate the risk of bias. So if you see pre-intervention, during the intervention, and post-intervention, there are 3 domains under which you have subheadings. There are 7 domains in total, and a 34 extensive 34 item checklist is there. But since we are not going to do a meta-analysis, but if our observational study in future, it can become an included study in a meta-analysis. So when the person who's going to do a meta-analysis is going to critically review the study, and if it is an observational study, he is going to use Robin's eye. So Robin's eye is another more deeper tool which is going to calculate, not just identify biases, but is going to calculate the risk, and this risk calculation is done in percentages. So maybe, for example, a selection bias, for example, bias in the selection, if there is no eligibility criteria mentioned in your study. So How much is the bias? Maybe 10%, 20%. This answer is going to be given to you by Robinson. Now we move forward. So more robust is your study design, less are the number of biases. So it is important, imperative on the author's part that we identify the sources of biases and we attempt at reducing them. So the reviewer actually wants to see in your manuscript what are the attempts made by the authors to reduce biases. So now let us go deeper into what is errors and biases. Error is something that is maybe humanly or technically or any instrument which you are using, and the error gets introduced. But if you allow, if it is, so that is unintentional. Actually errors are unintentional, but if you intentionally allow maybe missing blinding or maybe calculating the sample size, so that is an intentional attempt. So you are allowing these biases. Some biases get introduced, some are allowed by the researchers. So this slide, I'm not going to go deeper into what are errors type 1 and type 2. But if you see this situation over here, where a building is on fire, and here is the fire team, the rescue team, for first they are going to do two things here. One is save. The most important thing is save these people who are stuck in the building, and second thing is to douse the fire. So now there is going to be a decision maker. Suppose if that decision maker is you have to make two decisions. Either you evacuate or you do not evacuate. Okay. If the person, the decision maker, that is you, if you decide to evacuate, you are going to cause maybe a little bit of inconvenience to the others. I mean, if this is a dousable fire, it is good. But there might be elderly population who are living in the high floors, children and others, and the fire gets easily doused. Maybe the reason was a short circuit or something. But you did cause inconvenience to the others if the fire got doused. So because you chose to evacuate everyone in the building. This is an example of type 1 error. Okay. But if you choose to not evacuate the building and you continue dousing the fire, you are causing inconvenience, that is for sure, and the fire is spreading, but the risk to life is much, much more. So this is an example of type 2 error. Type 2 errors are intentional. Okay, when the authors allow type 2 errors to enter, and they are always dangerous for your results. So type 1 error is still okay. If you are identifying them, it is fine in your manuscript, but if you're allowing type 2, the results become shady. Going to biases. Biases, now once we know what is error, biases is when you deviate from the truth. You know what is the truth, or sometimes you don't know what is the truth when you're arriving at the results and the conclusion, but you still have a doubt and you start deviating from the truth. That is called as bias. Now this deviation comes into picture during data collection, when you're analyzing, when you're doing interpretation of the results, and when you report them. So actually you have collected something else, you're analyzing maybe around the same objective, but you are not giving a proper interpretation and reporting a third leg or a third arm of the trial. That is called as deviation. So this deviation is very much unethical, unwarranted, immoral, and misleading. So it is every researcher's responsibility to be aware of the potential sources of bias, and not that I did not know. And what action have you taken in your manuscript? What efforts did you make so that you do not deviate from the truth? is more interesting to the reviewers and future researchers, so they know what not to do. And that is how studies are more accepted for publication, if you stay on the line of truth. But if you are not giving answers, not answering the item checklist as given in Strobe and Petro and Robbins' eye, this means you have not done it. It is as good as saying that you have not taken that step in your management. So the sources of biases, like from where do these biases arise? If I just go a bit in detail, as we know, it is a responsibility of the author to be aware, first of all, and from where do they occur and arise. Because this is called as abusing data or torturing data. Now, if you can see this picture here on the bottom right, where the bear is trying to fish wherever he is moving the rod constantly to the right and left. So wherever he finds a fish, he will fish for the fish. So in research methodology, this is called as p fishing. P fishing is that we are so over dependent on the significance of p value that we arrive to our results or interpret our results in such a way that our p value should come significant. We don't like non-significant results. So this is a big problem. If you know that because of this, there are many journals which have come up who report negative results or non-significant results. Like you can see on the last line, Journal of Negative Reporting in Biomedicine. So they want you to report the results as it is and not because only significant results are published. But this is a misconception, it's a myth. Sometimes we avoid reporting limitations. We say that if we tell people or the readers our own drawbacks, maybe our research will not get published. In fact, people want to know, the readers want to know what problems did you face at every step of your research, because your methodology should be replicable. Okay, so instead of avoiding to report limitations, we should report limitations very comprehensively, in detail, in fact. Now, an example is of selection bias, I will say. How was the recruitment done? So sometimes I have few examples for you. Like patients were enrolled, maybe 100 patients were enrolled, but did you randomize them? Did you not randomize them? Did you enroll them by word of mouth or putting up posters? So if 156 patients were enrolled, then the eligibility criteria was applied and 55 were finally included in the study. So this number is very important, that it should be 55 out of 156. Okay, what was the screening procedure did you do? Many of the studies that directly mentioned 10 was the sample size, 20 was the sample size, and how did you come to this 20? So that becomes very shady. Okay, and then you cannot generalize those results to a larger population. Then it will stay to only those 20 results, and then we cannot do anything about that. Also, about dropouts. Dropouts is sometimes when you're calculating your sample size, in the sample size calculation, usually a good statistician or a good researcher will always foresee a 10% dropout. Always. So maybe if the sample size is coming to 25 or 20, maybe, after you have calculated your sample size of using the formula, plus minus 10%, you have to take at least more than 10%, like maybe 10% of 20 is 2. So I will take 22 samples for my study, foreseeing in the future that 2 might drop out. Okay, and so these are the reasons which are expected in a methodology, because it is not possible that if you recruit 20 or if you enroll 20 and all the 20 continue with the same procedure that you wanted to maybe do on them. There is a higher chance that some person can drop out, a person can die, a person does not want to do, can abandon the study, there is a loss to follow up, is not willing to give consent. There are so many reasons. Mention all of them. Okay? And that is how these are all small steps, baby steps towards making your methodology robust. The next is data inadequacy. Sometimes there are poor measurements. Sometimes you are so confident that the technology or the instrument that you're using is going to give you accurate results. Maybe it was not calibrated enough, at 0. So poor measurements. And if there is a chance of poor measurements, why don't you take 3 measurements and take an average of it? So that you have established the intra-rater reliability. Mention that in the manuscript or in your methodology, that my data is adequate. Sometimes you lose data. Sometimes even after following the intra-rater reliability, even if you have your data, the data which you receive, maybe they are images, X-ray images, CT images, or anything, blood samples, but they come out to be of poor quality. Mention that there were 2 samples which had poor quality and therefore I have excluded them. But you have already taken a +2 in your sample size adjustment. So this means that we can generalize the results. This is how research happens, and this is what people want to know, that what could be the problems that future researchers could face, and handling of missing data. How did you handle it is very important, because all these are pointers of Strobe and Robbins' eye. Okay, so moving forward, I'll just put on the laser pointer, sorry. So all in all, the last line is just report. Whatever you do, just report it. Okay, and report all sources of bias. So we have, in short, discussed the sources of biases. Now I will give you all examples. Like for example, if you see in this study, it's written that 30 participants over the age of 18 years, they were included in the study. But if you see the results on this side, so if you see the results on this side, they have reported males 18 and females as 16. So you cannot say that you have included 38 participants, but then you're dividing them into males and females. If the objective of the study is to study few characteristics in males and females, each group should have 38 participants, and then you apply a t-test. So males should be 38 and females should be 38, and not that after you divide them into 18 and 16 or whatever is the calculation in math. So this is an example of selection bias. If you see here in the study below, which I have highlighted, if you read the statement, so well they have written, only 6 subjects were excluded from our study owing to edema of venous insufficiency. There were 6, means 2 out of 6 were due to venous insufficiency. 1 person had diabetes, 2 person had diabetes, and 2 had femoral and tibial fracture. So they have given a very clear-cut methodology that how they excluded 6 subjects or whatever was their total. Okay, so this is these are very believable, means the results are going to be very believable. If we move forward, you look here how the study considerations and limitations have been extensively described, just like a discussion. Every point, whatever was in the methodology, the limitation part has been described. So you have studies which report that this difference was not statistically significant. They were not correlated. It is okay to use these terminologies. You see how the sample size is very much given in detail, what is the percentage of statistical power you used, how much was the beta error, how much was the alpha error level kept, because you can introduce the alpha and beta, that is type 1 and type 2, to a certain level and not beyond that. So what was the level did you set? And then the estimated number of participants was 28. So a very well planned study. The last slide of my presentation is what are the types of biases. So we have already, I've already discussed the types of biases, but not with their nomenclature. So whenever there is the source of bias is in the recruitment, randomization, or subgrouping, as we saw males and females, it is a type of selection bias. Whenever you use students or healthy volunteers between 20 to 30 age group, but you are recruiting the 20 to 30 age group people, volunteers from a college or a university, this means they are students, and students are vulnerable population. So that becomes a participant bias. Sampling bias, we have already seen when you are calculating the sample size. And if you don't mention or calculate the sample size, but you have calculated and not mentioned, it is a form of bias, sampling bias. All these biases, mind you, are part of the methodology only. Okay, the initial one. The next is the attrition bias, where you are comparing between two groups, but follow-up is not there. Okay, so it is actually just like the male-female example, a form of selection bias only. Basically, you have not done a good comparison of the independent groups in the beginning only. So then, and then you decide your own groups that it should be this way or that way. Form of selection bias, also called as attrition bias, if you want to be specific. Detection bias. Now, blinding. Sometimes if you see, not all, only just the question that did the author or researcher use blinding or not. That is only one part of detection bias. This blinding can be at 3 levels. The first blinding is when the assessors are blinded, the primary assessors. The second is when the participants are also blinded. They don't know what treatment they are going to take. And the third is when the outcome or the data that you get, that also can be blinded. It can be coded and then can be given for further processing. So all these 3 are part of blinding. Also detection bias. Statistical bias is, as I said, p-fishing. Sometimes you do not set a null hypothesis and you bypass it, or you use an inappropriate test. I mean, that is the most common thing, but setting a null hypothesis is very, very important in the beginning of your, when you are stating your aim and objective, and at the same time you state that this is my null hypothesis. Okay, but if you don't state and if you just report the results, so this means there can be no yes or no answer. Okay, when you conclude your study, it is going to be all in the air. That is a form of statistical bias. Reporting bias is when you have outliers, when you're trying to fabricate. Outliers means sometimes the standard deviation or the confidence interval or the variance in the variables are too at the extremes. Okay, identify them, think and discuss. In the results, you're going to identify the outliers, that yes, the standard deviation, yes, the variance did come high. What could be the possible reasons? Okay, report them in the discussion. They are all sources of reporting biases. Funding biases when the study is funded. Okay, usually the funders, the sponsors, the organizers want specific results from the authors. So a bias is introduced. Okay, publication bias is an example of meta-analysis, systematic review and meta-analysis, where not all studies which actually should have been in the systematic review are reported. So you have to actually go and search back the gray literature or manual hand search or other databases where this similar studies with the objective which you have for systematic review are there, so that you do not miss anything and you do not get a false positive result. That is publication bias. And sometimes the researcher can be biased. The researcher can be biased sometimes there are 2 groups and the researcher decides, or the patient convinces the researcher that I want to be in the second group, and the researcher allows it because the researcher is biased. So a researcher should have a neutral perspective when recording data, when segregating data, and actually during the time of data collection, and when you maybe interpret the coding the data or the images, you should have a very neutral perspective. You can have any answer. Okay, it is not necessary that it should be increased or raised, or it should come till a certain level. Okay, so biases are also variants of confounders or confounding variables. And with this, I end my the topic. So all I want to say is, when you're researching, you are a researcher first, and then you are maybe a doctor, an engineer, or whoever you are. So don't go into the details that this should come this way. Okay, you just research and report. Thank you so much.
Yeah, thank you, Dr. Sumaya, for your presentation.
Thank you.
So any questions? Okay, no questions. Thank you once again for your presence and presentation.
Thank you. Thank you.
Yes, so our next presenter is Dr. Shima Tariq Ahmed from Alexandria University, Egypt. And the title of the talk is Oxidative Stress and Antioxidant Defense System Among Workers of the Printing Industry. Hello, Dr. Shima. Hello.
Hello, Jessica. Hello, everyone.
Yeah, hi. So you are the presenter now. Please share your screen.
Can you hear me?
Yes.
Yes. Okay. Okay. Good morning. First of all, I would like to thank you all the management of the conference to let me share, and
Yes,
thank
now
you for your organization. Okay. My topic is about oxidative stress and antioxidant. I'm sorry. Oxidative stress and antioxidant defense system among workers of printing industry. I'm a chemist working in printing industry, and this topic is just a part of my postgraduate PhD. Postgraduate PhD. I'm sorry for that. Introduction. Printing industry is one of the largest and most geographically diverse manufacturing industries in the world. The work environment of printing industry usually contains a large number of chemicals, inks, alcohol, cleaner, solvents, and that are commonly emit high levels of volatile organic compounds, which may be inhaled or absorbed by the body, where the volatile VOCs are have a high vapor pressure and easily penetrate the unsaturated fatty acid tissues. Increased emissions of VOCs and their resulting impact on air quality are now considered to be a measure of environmental concern. As a chemical species with varying chemical reactivity, VOCs are very difficult to control in the presence of oxygen hydroxyl radicals and UV radiations. VOCs can easily participate in the series of photochemical reactions, formating smoke as final product in the presence of nitrogen oxide. Secondary contaminants such as ozone, aldehydes, nitrates can also be easily generated. The worker in the environment of printing industry is handled with printing ink, thinner, fountain solution, isopropyl alcohol, cleansing solution, and that all chemicals are source of VOCs, which contribute to air emissions. VOCs are source initiate free radicals in our body, which could initiate a condition of oxidative stress, either induced hematotoxicity, neurobehavioral effects, hepatotoxicity, or renal toxicity. Our objective was to examine the effect of chemicals used in the printing industry on lipid peroxidation and antioxidant enzyme in printing workers and blood samples. Material and method. The experimental design of this study contained two groups. The first group was randomly selected at local printing company at Alexandria City, Egypt. The first one is control 26 male healthy volunteers, donors of different age, 6 socioeconomic who are working in the other divisions of the same factory were classified into control, and the second group is the worker group. They are 26 male volunteers workers who also has been exposed to chemicals for a long time, 10 years around. The results? It was significantly noticed that the first the blue column represents workers and the orange column represents control. The lipid peroxidation biomarker was significantly increased in worker samples as compared to control, while the antioxidant catalase, superoxide dismutase, and the glutathione transferase and the glutathione peroxidase and the glutathione reductase were significantly decreased in the blood samples of workers as compared to control. Discussion. All types of organic solvents are lipophilic volatile liquids at room temperature. Adverse health effects related to exposure to organic solvents through inhalation and the skin contact in workplace. Exposure of solvent vapors affect not only the lungs but also the whole circulatory system and hence the whole body. Solvent accumulate principally in lipid fat-rich cells in the nervous system, brain, bone marrow, liver, body fats. Long-term effects may cause damage to the internal organs after absorption into the body. Lipids such as free and ester forms of polyunsaturated fatty acid and cholesterol are vulnerable target to oxidation by both enzymes and non-enzymatic oxidants. Solvents involved in the generation of reactive oxygen species that were found to induce cell damage through the increase and level of lipid peroxidation, decreased activity antioxidant enzymes, and generation of free radical. This could be conclude that work in play printing industry exposed to VOCs initiate a state of oxidative stress. This is result from an increase of production of active oxygen radicals or from inhibiting the ability of the antioxidant. Thank you for all. This study was under supervision of Prof. Dr. Fatma Demirdash and Prof. Dr. Huda at the environmental study Department Institute of Graduate Studies and Research, Alexandria University, Egypt. Thank you. Any question?
Thank you, Dr. Shima, for your presentation. So any questions?
Thank you. Thank you very much.
So no questions. Thank you once again for your presence and presentation. Yes. So as our next,
Thank you.
thank you. So as our next presenter has not yet attended, we'll go to the next upcoming next, and that is from Dr. Chunzi from Guizhou Medical University, China. Hello, Dr. Chun. Hello. Hello, Dr. Chunzi. So I will try to contact him. Please stay connected and allow me for a few minutes. I just contacted Dr. Z Shun, so she said that she will be joining in few minutes. So please stay connected.
Can you hear me? Jessica.
Yeah, hi, Dr. Z. Yes, we can able to see your screen. And please start presentation.
Good afternoon, everyone. It's my turn to give my report. I'm Xie Chun from Guizhou Medical University of China. My topic is the microRNA 34B5P target gene AI2 aggregates fluorine combined with aluminum induced apoptosis of offspring hippocampal neurons and NG108-15 cells. The directory includes 4 parts. As we know, appropriate fluorine is good for health, but excess of it can lead to fluorosis, which manifests skeletal fluorosis or dental fluorosis and non-skeletal damage, such as learning and memory impairment. Animal experiment and epidemiological survey have proved that excess fluorine can lead to learning and memory ability impairment. Aluminum is widely used in many fields, such as food pruning, cooking, and so on. It can cause changes in the nervous system and so on, and can lead to cognitive dysfunction and so on. Animal experiment and epidemiological survey have also proved excess aluminum can lead to learning and memory impairment. In the environment, there are fluorine and aluminum coexisting, such as fluorine and aluminum combined pollen area where choline is mixed with coal in Guizhou province, industrial waste air pollution area, and briquette type fluorine combined with aluminum disease area. Fluorine and aluminum can form complex. The complex can pass through the blood-brain barrier and lead to impaired learning and memory, but the mechanism is not clear. We have found microRNA 34B5P is one of the microRNAs differentially expressed in the hippocampus in offspring exposed to FA. microRNA 34B therapy participates in apoptosis and other cellular activities by targeting mRNAs. We predict the target gene of microRNA 34B therapy and found gene AI2. This gene is related to learning and memory, and its learning and memory related pathway is PKA-ERK-CREB pathway. The pathway regulates the transcription of genes and inhibits neuronal apoptosis involved in memory formation and consolidation. So we guess FA may be impaired learning and memory by apoptosis mechanism, and the mechanism is related to microRNA 34B therapy to regulate gene AI2 and PKA-ERK-CREB signaling pathway. So in this study, we want to verify the target relationship between this microRNA and gene AI2, and to know this microRNA expression and apoptosis in ratcheted NG108-15 cells exposed to FA, and to explore the results of microRNA 34B therapy in hippocampal neuron and NG108-15 cell apoptosis induced by FA. The animal treatment and the cell culture as Table 1 and Table 2. Research methods includes predict target genes of microRNA 34B therapy by these three soft, and use Go analysis and KEGG analysis, the function and pathway of microRNA 34B therapy, and to verify the target gene of it. The expression levels of microRNA 34B therapy and gene AI2 assay in ratcheted cell model is tested for mRNA and protein by qPCR and WB. Hippocampal neuron apoptosis was assayed by tunnel staining. Gene and protein expression levels of this microRNA, gene AI2, and this pathway by qRT-PCR and WB. Apoptosis was assisted by low-sat flow cytometry. Gene and protein expression levels of this MIR, gene AI2, and this pathway were analyzed by qPCR and WB. We have proved microRNA 34B5P regulates gene AI2 expression. Apoptosis of hippocampal neurons in each group exposed to FL and FA concentrated in DG. The percentage of apoptotic cells and index in all experimental groups were increased. In the hippocampus of offspring rats exposed to FL and FA. Compared with the control group, the mRNA expression of this micro and the gene AI2 in the hippocampus was increased and decreased respectively. Compared with the control group, gene AI2 protein in all exposure groups except the non-F group was downregulated. Compared with the control group, the mRNA of pKa and ERK and CREB was decreased many in most experimental groups in the hippocampus of offspring rats exposed to FAL and FA. The protein expression of pKa, ERK, and CREB in the hippocampus of offspring rats exposed to the exposed group many decreased. Apoptosis of cells treated with FAL and FA were increased. mRNA of this micro gene AI2 in cell models decreased or increased. The mRNA of pKa, ERK, and CREB in cell model decreased. The protein of gene AI2 was decreased. pKa, ERK, prophosphorated ERK and CREB and prophosphorate CREB decreased in cell models treated with FAL and FA. The apoptosis rate was increased in cell models treated with FAL and FA after upregulation of this microRNA. The mRNA of this microRNA was increased and the gene AI2 mRNA decreased. When cells treated with FAL and FA after upregulation of this microRNA, the mRNA of PK decreased. The mRNA of ERK and CREB increased. In cells treated with FAL and FA after upregulation of this microRNA, the protein of GNAI2 decreased. pKa, ERK, CREB, and phosphorylated CREB decreased. In cells treated with FAL and FA after upregulation of this mRNA, the only apoptosis rate and the total apoptosis rate decreased. In the no F group, medium F group, no FA group, and the medium FA group, and increased in the other experimental groups. When the cells treated with FAL and FA after inhibition of this mRNA, the mRNA of microRNA 34B5P was decreased and the gene AI2 mRNA increased. When cells treated with FAL and FA after inhibition of this mRNA, the mRNA of pKa increased, mRNA of ERK and CREB increased. When cells treated with FAL and FA after inhibition of this mRNA, the protein of GNAI2, pKa, ERK, and phosphorylated ERK, CREB, and phosphorylated CREB increased. When cells treated with FAL and FA after inhibition of this mRNA, the results indicate that damage mechanism of FA2 brain involves apoptosis of hippocampal neurons and microRNA 34B5P is closely related to NG100 and H15 cell apoptosis, and that overexpression of microRNA 34B5P can promote cell apoptosis. Continuous exposure to FA causes a decrease in learning and memory due to overexpression of microRNA 34B5P and downregulation of gene AI2 expression. Compare the results of this study and other studies. We can know the conclusions. FA can induce apoptosis of offspring hippocampal neurons and NG188 cells. MicroRNA 34B5P targets and regulates the expression of gene AI2. FA can upregulate the expression of microRNA 34B5P and inhibit the activation of the gene AI2 and PKA-ERK-CreB signaling pathway, which may be one of the mechanisms by which FA exposure leads to neuronal apoptosis. This is Guizhou Medical University, a beautiful university. Welcome to our university. Thanks for your listening.
Thank you, Dr. Zishun, for your presentation. So, any questions?
This is my email. If you have any question, please contact with me.
Yes, so if you have any questions also, you can directly send the questions to the mentioned email ID, or you can send it to us. We will forward it to Dr. Chunzi. So thank you once again for your presence and presentation.
Thank you for giving me this opportunity.
Yes, thank you very much. So by this, we came to end of our day 2 webinar as well. I would like to thank all the attendees and participants of today's webinar. Hope you enjoyed this and gained some knowledge from this session. And we will be glad if you could join us in our next in-person event, which is scheduled on November 14 and 15 in Dubai. The conference on the pharmacology and toxicology. So regarding the details will be sent to you an email, and the certificate will be sent to you in short while after the conference is going to complete. And also we will be very happy if you could share your opinion or feedback with us through email. So thank you one and all. Thank you. Very happy.
Thank you.
Have a nice day.
I'll end this share. Okay.
Yes.
Okay.
Thank you. Have a nice day for you.